Chapter 19 is about viruses, and it's kind of a unit on itself, so I'm just going to throw it on the end of the genetics. Sometimes it's left out, but considering I'm teaching this way because of a virus, I figured, yeah, we better talk about viruses. So chapter 19 is about viruses. Viruses have a borrowed life. They cannot survive or function alone. They have to basically get inside of a host cell, take over, and then that's how they live. When they're not inside of a cell, they don't do anything. Really, what a virus is, it's a piece of either DNA or RNA, so it's a nucleic acid, one gene usually, and it's protected by like some sort of protein coat. Okay, they're very simple in structure, even simpler than prokaryotic cells. They're much smaller, much simpler than bacteria. Okay, and they cannot reproduce. They can't reproduce. They can't carry metabolism. They basically can't do anything unless they live inside of a host cell. So that's their job, to get inside a host cell and take it over. Okay, they're parasites basically. And here's a picture you've probably seen a lot of virus pictures with the coronavirus, right? Again, they detected them long before they could see them. And the reason for that is the microscopes were just too tiny. They had microscopes that were good enough to see bacteria, and they knew things were, getting diseases, and they couldn't see any bacteria, so they knew there was something else causing them. I don't know when they decided to call them viruses, but really, again, a virus is just a nucleic acid, a little piece of DNA or RNA, and it's protected by a protein coat. So the disease that they first got the idea that there's something smaller than a bacteria causing it is tobacco mosaic disease. The tobacco plant doesn't grow very well, and the leaves get discolored. They have many colors in them. There's where the mosaic comes in. And tobacco was a huge industry when the country first started. So, you mess with the tobacco, and they got to figure out what's going on because it's important to the economy. So in the late 1800s, they started saying, we've done and we've tried and we cannot find a bacteria that's causing this disease. So there must be something even smaller than a bacteria. And the guy that actually confirmed that these little tiny disease-causing creatures lived was a guy by the name of Wendell Stanley. 1935, so less than 100 years ago. He worked with the tobacco mosaic virus, the TMV he called it. He knew it had to be there, and he had an experiment, and this is how he proved it. He took a plant that had the virus, and he churned her all up and put her in the blender, and he poured it through a special filtration system, and this has a porcelain filter. This will trap all bacteria. Okay, so if there was a bacteria causing it, it would be up here, not down here. Well, then he took some of this stuff and he put it on a healthy plant, and sure enough, it got the disease. So he knew that there was something even smaller than a bacteria that caused it, and that's where they got the name viruses. So they knew about it way before they could see it. Okay? Again, remember, a virus is not a cell. It's a very small infectious particle. It has a little piece of DNA or RNA. It's protected by a protein coat. Sometimes it even has another envelope over there. It depends on the virus. Okay? So viruses aren't just the same thing. We have different viruses. They have different structures to them. Some viruses actually have accessory structures to help them get inside the host. Some sort of, hook or something to help them so they can get inside. Because remember, we have this membrane that covers the cells. Well, they have to get through that in order to get inside the cell. So they might have some sort of an accessory, depending on the virus, to help them get inside the cell. Okay, sometimes they have an envelope, a viral envelope they call, and it actually comes from membranes of their host cells. So they're borrowing some membrane from them. So maybe when they come to another cell, it'll recognize them, as a different, it won't recognize them as a virus, but instead as a cell membrane because they've borrowed some of their stuff. The influenza virus, the one that causes the flu, tends to do that. Okay, so viral envelopes contain a combination of the envelope from the virus, and they've borrowed a little bit from their host cell. And again, that helps them get recognized so they can get inside of a new cell. Okay, viruses replicate only inside the host cells. Okay, a virus cannot reproduce unless it is inside of a host cell. Okay, so before it gets in a host cell, it's, that's why we use all our sanitation things and the wipes and everything, because if the virus is on your table, you wipe the table, you kill the virus. The only way the virus works is to get inside a host cell and then take over. Okay, They have a range. I mean, it isn't like every virus can infect every host cell. There's only certain ones that can infect. Okay. There's some diseases that only affect pigs and cows and horses and whatever. So not every virus can affect every cell. Okay. So once that genome, again, that's your little piece of DNA, once it gets inside of a cell, what it does is it starts to take over. It starts to produce its own proteins, and by using what was already in the cell. So it uses the cell's factories to make their proteins. Okay. So it's kind of crazy, but it's a good idea. Go in there and take over and let them do all the work. And you don't have to have all that material. Well, you don't have room for it. Okay. And they make, again, they use the ATP, they use the transfer RNAs, the amino acids, everything that's already there. They just, instead of sending the messenger RNA coming from the DNA, they send their RNA or their DNA. Okay, so they steal the information, if you want to call it that. Okay, so vironucleic acids, molecules, they call them capsomeres. Sometimes they can spontaneously self-assemble into new viruses. This is why we can never have a cure for the common cold, because cold is caused by the rhinovirus. Okay, once you get the rhinovirus, you shouldn't get it again because now you have antibodies, immunities, all that. But it changes. So then when you come in contact with a new one, you get a cold. And then you get over that one, and then you think, I'm good now. No, you can come in contact with another one. So that they change. Not all viruses do, but many do. Okay, here's a picture. We got our little virus. It's really just a little piece of DNA with some sort of covering on it. It gets inside the host cell, and now it starts making its own proteins. They're called capsid proteins, okay? These aren't supposed to be there, but they trick the cell into making their new ones. And so then you end up with a whole bunch of new viruses. Okay, so it uses the host cell to produce more viruses. Okay, there's all kinds of viruses. The one that they know the most about are called phages. A phage can have two different types of cycles, the lytic and the lysogenic. It can go in between the two of them too, which is kind of a pain. But the lytic is going to destroy the cell. It's going to produce a whole bunch and then kill the cell and open up and release them all. And the lysogenic is going to insert its information, and so that every time the cell divides, it has that information in it. So they have two. Okay, so again, the lytic cycle, what happens is the phage gets inside the host cell and it produces a bunch of babies. And then when there's enough babies there, it kills the cell and releases all the babies. And so then they can go find more host cells and so on and so forth. Okay. It produces new phages. And then again, it breaks open the host cell wall or the cell membrane and releases its babies, its progeny into the, and now they're going to go out and they're going to look for new cells and do the same thing over and over. Again, we have some things to stop that, but for a little while, this is kind of the incubation period, and I know you guys know what I'm talking about. When you know you're going to get sick, but you're not quite sick yet, that's what's going on. Okay, the phage that reproduces only during the lytic cycle is called a virulent phage. So there's some that only do this. They get in the cell, take it over, and then send their little babies off to do more work. Okay, there's some bacteria that have defenses against phages. We have cells in our body, our white blood cells, that chomp them up. You know, when they release those little phages, they eat them. That's why when you have an infection, your white blood cell count goes up. Okay, so there's certain ones that they can chomp up, and that's really how you get better when you don't die of a cold, so something has to take care of those little phages. Okay, the lysogenic is a little bit different. What it does is it replicates the phage. It inserts its DNA basically into the DNA of the cell that's already there. So every time that cell reproduces, now it has new DNA in it, and the new DNA came from the virus. So it incorporates its DNA into the DNA that's already there. Okay, and it's this is called prophage. Okay, the integral viral DNA now becomes part of it. So it inserts its DNA into one of the chromosomes. And again, every time that cell divides, now it's got new DNA with the virus DNA in it. Okay, so it passes it along. Okay, now sometimes something in the environment can trigger that cell that's in the lysosomic and say, oh wait, this isn't working. Now we got to switch back to the lytic, so it'll take over and it'll destroy the cell and release its babies. Okay, some viruses, only do the lytic, some only do the lysogenic, some do both. It depends on the virus. But they're usually, when they're in the lysogenic, it's there, and it's just something's going to happen to it to make it switch. There's some sort of environmental trigger that happens to it. Okay, so phages can use both. If they do both, they're called temperate phages. It means they can switch between them. So they can do this one, they can do this one. If they do both of them, they're called temperate. Okay. When we're talking about the cycles of animal viruses, okay, there's two variables they look at to classify them. Okay. They're going to look and see, is it an RNA or a DNA? Okay. So when we're looking at viruses that infect animals, we're going to see, is it a, because I said virus is a nucleic acid with a protein codon. Is it an RNA piece or is it a DNA piece? Okay, is it single-stranded or is it double-stranded? Well, if it's an RNA, it's going to be single-stranded. If it's DNA, it's going to be double-stranded. Okay? And then we're going to look at these bacteriophages. Okay, do they have an envelope around them? Okay, do they have an envelope or do they have an RNA genome? And again, it makes a difference to what they can do. Many animal viruses really have an envelope and the RNA genome. They have both. Okay, not all, but a lot. The broadest variety of RNA genomes is found in viruses that affect animals. Okay, we have viruses that affect plants. We have viruses that affect, there's all kinds of things that are infected by them. But when we're looking at the viruses themselves, there's a broader variety in animal ones than there is in plant ones. Okay, then we have something called retroviruses, and these weren't really big. They didn't know much about them until HIV. Okay, it took a long time to isolate what HIV was because it's not a phage, it's a retrovirus. Okay, they use reverse transcription to copy their RNA genome to the DNA. So remember, if it's RNA, that's supposed to be, copied from the DNA. Well, in this case, they do it opposite. They take their RNA and they copy it into DNA. So that's why they're called a retrovirus. They're doing it reverse. Okay, and again, the biggest retrovirus, the one that people know the most about, is HIV. It causes acquired immune deficiency. Okay, so the HIV, the human immunity deficient virus, is a retrovirus. So it is a piece of RNA that actually copies it back into DNA, which is, like I said, kind of backwards of what we're used to. Okay, the viral DNA that is infected in the host genome is called a provirus. And then it's not like a phage, okay? It remains permanent resident of the host cell. A phage will either take over the host cell, have its baby kill the cell and be done, or it'll become part of the DNA, but it doesn't stay in the host cell forever. Provirus does. A retrovirus will get in the host cell and stay there. It'll become a permanent resident. Okay, so then it copies its RNA into the DNA. So again, it does everything backwards. Kind of crazy, right? Okay, so the RNA molecule functions both as messenger RNA for making new proteins, and then it also serves like DNA to make copies so that particles can be released from the cell. So it's backwards from what normally happens, okay? Normally we have the DNA molecule is copied and make messenger RNA and so on. Well, they already have their messenger RNA made to make proteins, and then they can make another copy, a different type of RNA from here to make other proteins. So they can actually make two, whereas most phages can only do the one thing because they only have that piece, but these guys can go both ways. So that's why retroviruses are different, and they're harder to study, they're harder to isolate because of the things they do. Okay, I think I'm going to quit there, and then we're going to do this chapter in just 2 parts. It's not a long chapter, and again, we'll get her done.